Hello viewers, welcome to Mesology. In this video, I will try to give you a clear idea about the chromosomes, DNA, genes, karyotype as well as the genome. Let us get started with the two basic kinds of cells. The first are the prokaryotic cells, the cells that are found in a bacteria, that is called a prokaryotic cell. These prokaryotic cells contain a single length of DNA. This DNA lies inside the protoplasm, which is often referred to as the nucleoid. But generally, when we talk about chromosomes, we often refer to the chromosomes which is found inside the nucleus of an eukaryotic cell. Now, we should first know what an eukaryotic cell is. So, eukaryotic cells are the cells that are found in your body, in the cells of a plant, or that of a fungi or a protist. When it comes to the eukaryotic cells, these organisms have chromosomes which are located inside the nucleus, and the chromosomes are bound inside the nucleus by a nuclear membrane. On closely viewing such cells under a microscope, usually a compound microscope of high resolution, one can see some characteristic X shaped structures. These are called the chromosomes. It is true that these chromosomes are not visible even under a microscope when the cell is not dividing, as they remain in an uncoiled state called the chromatin network. Moreover, they are extremely thin in diameter to be viewed under a light microscope. However, in a dividing cell, the chromatin coils tightly, condenses, and eventually becomes visible. When stained, they readily get colored and become visible as darkly stained bodies. This is why we call them chromosomes, since chroma means colored and soma means body. They were first reported in the year 1882 by Walter Fleming in salamander larvae. Each chromosome has a primary constriction, which is called the centromere. This centromere serves to attach the chromosomes with the spindle fibers during cell division. It also helps to detach the sister chromatids during cell division. The location of centromere helps to provide the chromosomes a characteristic shape which is used to describe the location of specific genes. So, according to the position of the centromere, chromosomes can be metacentric when its centromere is almost at its center, producing two equal arms, the P arm and the Q arm. But if the centromere is little shifted away from the center, the chromosome is called submetacentric. So there are two unequal arms. We again have the other variety where the centromere is shifted almost at one terminal point. Such a chromosome is called a telocentric chromosome. On the other hand, if the centromere is a little shifted inside from the extreme end, it is called acrocentric chromosome. One vertical half of duplicated chromosome is called a chromatid. Humans have 23 pairs of chromosomes. And out of each pair, one is inherited from the father and the other is from the mother. So, a pair of these corresponding chromosomes of the same shape and size, one from each parent, makes a homologous pair. So, the pictorial representation of the human chromosomes lined up sequentially in pairs is called the karyotype. We can otherwise say that the karyotype is the picture of the full set of human chromosomes in a person's cells. Uncoiling of the chromosomes carry the following structures: a double helical DNA, which makes up around 40%, and the histone proteins, which make up a little more, and that is around 60%. In this context, we have several names of scientists who have worked to find out the structure of the DNA. Rosalind Franklin was the first to give the shape of a DNA molecule in the year 1953. Unfortunately, she was not awarded the Nobel Prize for the structure of DNA. However, the Nobel Prize was awarded to Watson and Crick for proposing the double helical structure of DNA in the year 1962. A single DNA molecule is very large, thus, it's called a macromolecule. Two complementary strands wound around each other in a double helix. Each strand is composed of repeating units called the nucleotide. Each nucleotide has three components: a pentose sugar, a phosphodiester bond, and a nitrogenous base. The nitrogenous base attached to the sugar inwards. It's a ladder like arrangement. The nitrogenous bases form the rungs of the ladder. Two nucleotides are found at each step of the ladder. Hydrogen bond binds the two complementary base pairs. There are four different base pairs, out of which adenine and guanine are purine bases, while the cytosine and the thymine are pyrimidine bases. One purine base complements with another pyrimidine base. It is adenine that pairs up with thymine, and the bonding between adenine and thymine is a double covalent bond, whereas cytosine pairs up with guanine, which makes a triple bond between them. A DNA strand winds around the core of histone octamer, forming a complex called nucleosome. In humans, there are more than 1 million nucleosomes in each chromosome, and there are 23 pairs of chromosomes in the nucleus of each cell, where each human cell contains around 2 meters of DNA if it is stretched end to end. However, the size of the nucleus is approximately 6 micrometers in diameter. So, the DNA needs to be condensed, and the entire chromatin fiber has to be coiled and supercoiled so as to fit into the nucleus. Now, where are the final unit structures of heredity? What we know are the genes. These genes are actually specific sequences of nucleotides. And these nucleotides are present on the DNA of a chromosome that encodes a particular protein which is able to express in the form of a particular feature of the organism's body. These genes are the units of heredity that are responsible for the transmission of different features from the parents to the offsprings. What we call it as inheritance of characters. Cytogenetic mapping of chromosomes give just a bird's eye view. With the advent of new techniques in the DNA analysis, much greater details were possible. The nucleotide arrangement in the DNA could be seen by the X-ray crystallography. Moreover, the human have 23 pairs of chromosomes and approximately 20,000 different genes on their DNA. But a single DNA is with a width of 2 nanometers and a length of 0.34 nanometer per turn. So the DNA is extremely small to be viewed under a light microscope where the visible range of light is between 400 to 700 nanometer. In the pre-sequencing days, chromosomes were identified by the staining pattern and the location of centromere. The autosomes, other than the sex chromosomes, are numbered by the decreasing length. That is, the chromosome number 1 being the largest and chromosome number 22 is supposed to be the smallest. But of late it has been found that chromosome number 1 is indeed the largest, which is around 3 times bigger than the smallest chromosome. But chromosome number 21 has been found to be smaller compared to chromosome number 22. Moreover, chromosome number 19 has also been found to be smaller than chromosome number 20. When we consider the entire karyotype, then there is a controversy between the Y chromosome and chromosome number 21 as to which one among these two are the smallest. Some of them claim that chromosome number 21 is the smallest. So, we can summarize that the gene is the smallest unit of heredity which is present in the DNA. Then this DNA is coiled along with the histones to make up the chromatids. The chromatids condense, coil and supercoil to make chromosomes. And finally, the full complement of DNA, including all the genes and the intergenic regions, make up the genome of an organism. The groundwork of the ultrastructure of chromosome was laid down by several scientists. Willem Johansen in the year 1909 coined the term gene, genotype as well as phenotype, whereas Bateson introduced the term genetics for the branch of science of inheritance of characters. There are other scientists, namely Albert Kozel, pioneer in genetics, who won a Nobel Prize in Medicine in 1910 for his work in determining the chemical composition of nucleic acids. Johannes Friedrich Miescher, he was a physician and a biologist to first isolate nucleic acids in 1869. It was then in 1909 Phoebus Levin and Walter Jacob they determined the deoxyribose as a natural product and essential component of nucleic acids. In 1929, he discovered deoxyribose sugar and showed that the components were linked together in order, forming phosphate, sugar, base units. Until 1900, these results were not understood. Erwin Chargaff, he researched into the chemical composition of DNA that laid the groundwork for Watson and Crick. Thank you for watching. If you have any queries, please do post it in the comment box. I will attend to it shortly. If you like my video, please do give a thumbs up. Share it with your friends who may find it valuable, and also do not forget to subscribe to my channel. Thank you once again.